All right, so today we're going to cover chapter 11, which is the sense organs, or sometimes referred to as special senses, but it's everything related to our senses. So mostly that's going to be our eyes and our ears, but what are the rest of our senses? So we have 5 senses, right? See, hear, taste, smell, feel. Like I said, eyes and ears are going to be the biggest part of it. We're going to talk briefly here in the beginning about some of the rest of it. But in a general sense, what do our senses allow us to do? They really allow us to know what's happening in our environment, which ultimately allows us to be protected from danger and injury, and know what's around us, what's coming, be able to taste our food. Is it food? Is it poison? Any of those types of things is what our sensory system allows us to do. So this is going to be a little bit of a review from neuro last week, but we have sensory receptors, right? We have millions of these throughout our body. Some combine with muscle and tissue to form sense organs, but most of them consist of specialized nerve cells or nerve endings that detect chemical or physical events outside the cell membrane. So here we have this sensory pathway, and this again should look really familiar. This is kind of the pathway that we looked at in our activity last week. So we have a stimuli here, right? Pain, sight, hearing, any of those things. And this is a receptor here, basically, or the dendrites of the neuron. And they receive the stimulus. And if the stimulus is strong enough, the receptor causes a sensory neuron here, okay, to send the impulse to the brain through the spinal cord, after which, we actually then experience this sensation, sight, sound, taste, whatever it is. The brain does a really nice job of filtering out most of the stimuli detected by receptors because we kind of want to keep them below our level of awareness. We would become really overwhelmed very quickly if we were alert to every little sensation that our body detected. So by keeping them kind of below our level of awareness, it allows our body to still respond to them, but kind of respond to them without us knowing. So it's important that our body does that, and we want our body to do that. There's some information we need to know about sensory receptors. So the type of stimulus, so each sensory receptor responds to a different type of stimulus. We have cold receptors, they respond only to cold. We have light receptors, they respond only to light. And then the location really allows us to pinpoint the location of the stimuli. So each sensory neuron responds to a stimuli in a certain area called a receptive field. Sensitive areas, such as the tips of our fingers, contain many receptors. Because each neuron only covers a very tiny territory, this actually is what allows the brain to pinpoint the location of the stimulus. Less sensitive areas, such as our back, contain fewer receptors. Consequently, the brain can't pinpoint the stimulus location as well. It really depends on what part of our body we're talking about and how pinpoint the brain can tell where that stimulus is coming from. The other piece is the intensity of the stimulus. So the stronger the stimulus, the more nerve fibers that are stimulated, allows the brain to interpret the intensity of a sensation. When a stimulus is continuous, the firing frequency of the nerve slows, causing the sensation to diminish. This is known as adaptation because we want to trigger our body to respond to that, but if we don't respond to it, usually our body will start to respond less to it. Sometimes in nursing, this happens with call lights or some of those frequent noises that we hear all the time because our senses are constantly being stimulated when we're in that type of an environment, and our body starts to kind of tune them out. So we have different classifications of receptors. We have chemoreceptors. They kind of detect chemicals, things like odors, taste, and the concentration of such chemicals as glucose or carbon dioxide. We have mechanoreceptors. They respond to factors such as pressure, stretch, and vibration that change the position of the receptor. We have thermoreceptors that are activated by a change in temperature. And then we have nociceptors. These are pain receptors that respond to tissue damage from trauma, as well as from heat, chemicals, pressure, or lack of oxygen. We have photoreceptors that respond to light, and they're found only in the eyes. And then we have proprioceptors. These are found in the skeletal muscles, joints, and tendons. And they provide information about body movement, muscle stretch, and the general orientation of the body. So those are the different types of receptors that we're going to find within our body, but also within our sense organs. So general senses, this is a general definition of the word general senses. That includes pain, pressure, touch, stretch, and temperature. They involve receptors widely distributed in the skin, muscles, tendon, joints, and viscera. Just kind of some of those things we just talked about. Really, this is everything outside of seeing, hearing, smelling, and tasting. And this is the kind of other piece that of our sensory system that still allows us to know what's going on outside of our body. Pain, again, like I said, nociceptors are what actually detect pain, and they consist of free nerve endings that carry impulses to the brain. We have fast pain receptors and we have slow pain receptors. The fast pain receptors are abundant in the skin and mucous membrane, and they produce a sharp, localized, stabbing type pain. Versus the slow pain fibers, they're congregated really in deep body organs and structures, and they produce a dull, aching pain. And just on another note, something interesting to note, the brain doesn't contain any nociceptors. We really don't get pain with brain injury, but we get pain because there's nociceptors still on the covering of the brain, the skull, those types of things. But actually when doing brain surgery and they're actually manipulating the brain, you're not actually going to get pain. Here's the pain pathway. I don't want you to memorize every step of the pain pathway, but I do want to show you the animation so you can see how this pain pathway actually works.
When an injury occurs, injured tissues release chemicals that stimulate nociceptors and trigger pain. A neuron conducts a pain signal to the dorsal horn of the spinal cord. At the dorsal horn, the spinothalamic tract relays the pain signal to the thalamus. At the same time, the spinoreticular tract carries pain signals to the reticular formation of the brainstem. The thalamus relays the signal from the spinothalamic tract to the postcentral gyrus of the cerebrum. At that point, the person becomes aware of the pain. Meanwhile, the impulse from the spinoreticular tract bypasses the thalamus and travels to the hypothalamus and limbic system. These areas trigger emotional and behavioral responses to pain, such as fear and nausea.
So you can see there how the pain pathway works. Again, I don't want you to remember all those steps, but I think it's just a good visual to see there of how the pain pathway actually works in people. The other piece that I want to talk about in relation to pain is referred pain. I mean, really, this is what this means is that pain originating in a deep organ may be sensed as if it's coming from another area on the body's surface. This diagram shows you different areas where people might be having referred pain. So sometimes people have heart pain, it's in the middle of their back. Sometimes people have urinary bladder pain, it seems more like it's in the rectum. Sometimes kidney pain you see here is actually kind of all over the place. So I think this one's really interesting. Liver, gallbladder, sometimes up in the neck. So it's really coming from the liver, but it's showing itself as if it's coming from the brain. Why this happens is because sensory fibers from an organ and those from an area of skin converge in a single pathway. Sensory impulses from both areas travel to the brain over the same pathway. As a result, the brain might misidentify pain from one area, such as the heart, as coming from another area, such as the arm. So because they ultimately converge on one pathway, the brain sometimes gets a little confused. Again, you don't have to remember, that heart pain, is here or there, any of these specifics, but I want you to know why it is referred pain. Why does referred pain happen? And that's because those sensory pathways converge and then the brain gets slightly confused. Special senses include taste, smell, hearing, equilibrium, and vision. And that's what we're going to spend the rest of this lecture talking about. The first one I want to talk about is taste. So you don't need to know all these different papillae. What you do need to know is that taste is known as gustation, And that results when chemicals come in contact with taste buds, most of which are located in protrusions called papillae. So you can see this picture here is showing us the taste buds here deep down in these grooves of the papillae. And really, the chemicals have to be dissolved in saliva. The saliva kind of allows us to actually taste. Because what happens is we get these little hair-like projections coming off of the taste pore, they call it. And those are bathed in saliva. So the chemicals dissolve in the saliva, and that's what actually stimulates the gustatory cells, which then stimulate certain cranial nerves and allow us to taste. So this is the tongue here, the little hair cells off of what we're calling the taste kind of pores, or the gustatory cell. And those are stimulated only when that chemical is dissolved in saliva or a solution. We have 4 primary tastes: sweet, sour, salty, and bitter. They're all scattered throughout the tongue, although some areas might be slightly more sensitive to different tastes. Smell is another one I want to talk about here. Again, you don't need to know the different steps that kind of happen here, but you need to know that smell is also called olfaction. So here it's coming off the olfactory nerve, so smell equals olfaction. And smell and taste are really related. You have to be able to smell to be able to taste. The other thing about the olfactory senses, or our sense of smell, is that they're quickly fatigued. So we really stop sensing smell after a pretty short period of time. What happens with this and how we actually smell things is we have incoming odor molecules here, and they bind to cilia projecting from the ends of the olfactory receptor cells. This stimulates an impulse along nerve fibers, leaving the nasal cavity through pores up through the ethmoid bone. The fibers synapse with other neurons and olfactory bulbs, and then we get the signals to go to the primary olfactory cortex in the brain. So that's the pathway to the brain. And again, usually these odor molecules are kind of suspended in air molecules, so that's how they're getting into our nasal cavity. So on to hearing. Hearing and vision are a little bit more of in-depth structures and processes that we need to really spend some time talking about. So the ears provide our sense of hearing, and they also provide our sense of balance, or our sense of equilibrium. So they do actually do 2 functions. There's 3 sections to the ear. We have the outer ear here, the middle ear, and then the inner ear. The outer ear consists of the auricle or the pinna, 2 different terms for that, really the ear lobe. Then we have the auditory canal that leads through the temporal bone here and into the eardrum here, or the tympanic membrane. That's the other word for it. The one thing that's important that happens in here is we have glands that produce secretions called cerumen, C-E-R-U-M-E-N, cerumen, which is known as earwax. And it really does have a job. It helps to waterproof the canal, and it helps to trap dirt and bacteria. We really shouldn't be sticking anything in our ear less than our elbow, so nothing smaller than your elbow in your ear, if you've ever heard someone say that. We often use Q-tips and stick them in there, but they often do a lot of damage, and our body can get rid of earwax on its own. Really, a hot shower with shampoo and the earwax actually melts and comes out with whatever we don't need. So just something to keep in mind. The middle ear contains the 3 smallest bones in the body. They're really named for their shape. So we have the malleus, which is the hammer, the incus, which is the anvil, and the stapes, which is the stirrup. And their job really is to connect the eardrum to the inner ear. And they allow sound waves to get to the inner ear where actual hearing sensation occurs. The stape fits within the oval window of the vestibule. So the oval window here is what actually transmits that vibration to the inner ear. The tympanic membrane here separates the outer ear from the middle ear, and it vibrates freely to sound waves. So this kind of starts the vibration. This vibrates, then these 3 bones vibrate and takes the vibration into the inner ear. The other thing that happens is we have the eustachian tube here, and that's a passageway from the middle ear to the nasopharynx. So everything's really connected. It equalizes pressure on both sides of the tympanic membrane, but it also allows for infection to spread from the throat to the middle ear. Much more common in children than adults, because in children this eustachian tube is very horizontal. In adults it actually tips up, so it makes it slightly more difficult for the bacteria to get to the inner ear in adults. But that's often why children have a lot of ear infections. The inner ear is where things actually get a little bit more complicated. So we have multiple things happening here in the inner ear. The inner ear is also known as the bony labyrinth. It consists of a complicated system of passageways within the temporal bone. So this here is showing you that it's actually inside the temporal bone, which is why it's called the inner ear. The bony labyrinth consists of semicircular canals over here, okay? The vestibule, which is this portion here, and then the cochlea, which is this portion here that looks kind of like a snail's shell. These semicircular canals are where equilibrium and balance occur. The vestibule really marks the entrance to the labyrinth and contains organs necessary for the sense of balance, so also involved in balance. And then here is where we actually have hearing occurring. So this is the inner ear continued, and this really is showing you more in depth down inside the cochlea where hearing actually occurs. So let's talk through what's really occurring here. So the spirals of the cochlea, if you can think back, looked like that snail shell. It's really divided into 3 compartments, okay? So here you can see the 3 compartments, 1, 2, and 3. The middle compartment is a triangular duct called the cochlear duct here, okay? That's filled with endolymph. And then the other 2 outer compartments are filled with perilymph, okay? And this is showing us down even a little bit, breaking this down a little bit farther. So this is down here inside the middle portion, okay, of the cochlea. Resting on the basilar membrane here of the triangular duct is the organ of Corti, okay? And that's actually the hearing sense organ. The organ of Corti consists of a layer of epithelium. Thousands of hair cells project from this epithelial layer and are topped with a gelatin-like membrane called the tectorial membrane, okay? So this is really jelly-like, okay? Nerve fibers extending from the base of the hairs eventually form the cochlear nerve, okay? So how hearing occurs actually is here on this slide, and this is going to show you actually how it occurs. So here, sound waves come in, they hit the tympanic membrane, that starts to vibrate, vibrates the 3 bones of the middle ear. That vibration goes into the inner ear where we have this endolymph, okay? And it vibrates, it goes, causes vibrations then to occur within the middle portion of the cochlea, and we get those hair cells moving in that gel-like substance, and that sends the stimuli to the brain, and actually what allows us to hear. So let's watch the animation on that, just to review that again.
Hearing occurs when sound creates vibrations in the air, known as sound waves. Sound waves enter the ear and travel down the external auditory canal. The waves strike the tympanic membrane, causing it to vibrate. The vibration spreads through the malleus, the incus, and the stapes, the 3 bones of the middle ear. The movement of the stapes against the oval window shakes the perilymph on either side of the cochlear duct. The ripples in the perilymph are transmitted through the roof of the cochlear duct to the organ of Corti. Movement here stimulates the hairs of the organ of Corti to send nerve impulses along the cochlear nerve. The impulses reach the auditory cortex in the brain's temporal lobe, where it is interpreted as sound. The ripples continue through the perilymph and dissipate by striking the round window.
So you can see how it's kind of a complete circuit. It comes in and then they dissipate as they strike that round window. So then they stop. Okay, so that's one big function that happens within inside the ear. The other is balance and equilibrium, and this occurs by 2 different processes, okay? So balance occurs both in the vestibule, and we have it occurring in the semicircular canals. So we have 3 fluid-filled semicircular canals, and they lie at right angles to each other. So one's up, one's here, and then one's kind of perpendicular to that. And inside the vestibule are 2 sense organs called the urticle and the saccule, okay? Those are two organs that are here inside the vestibule. We also have the ampulla here inside the semicircular canals, and I'm going to show you what occurs at both of those two places. So inside the ampule, we have hair cells that are again here, and we have the sensory nerve fiber. And these nerve cells are really tapped by this gelatinous cone-shaped cap. And this lightweight cap kind of floats in the endolymph that fills the semicircular canals. What happens is we get movement of this, and that actually is what tells us our position in space. So the ampules was inside the semicircular canals. Inside the vestibule, we have the otoliths. So somewhat similar, but a little bit different. So this is where we have the utricle and the saccule, and it consists really of a patch of hair cells. So the tips of the hair cells are covered again by this gelatin-like material, and embedded throughout the gelatin material, these heavy mineral crystals called otoliths. So again, we're going to get movement of the head, and these are going to move and they're going to stimulate the hair cells. What I'm going to show you is the difference between the two. So in the ampule really helps us to sense our dynamic equilibrium, or the speed and direction of head movements. So when the head rotates from side to side, the endolymph inside the ampule kind of lags behind. It's kind of heavy and gelatinous. When the movement stops, the endolymph swirls past that cap that's over the hair cells, bending it and pulling on the hair cells. This stimulates the nerve receptors, okay, to send a signal to the brain, and the brain interprets the information and triggers the response for maintaining balance when the head or body is suddenly moved. So this allows us to, again, keep our body upright when we move our head, doesn't throw our equilibrium completely off. This is why, or these two features are why, sometimes people who have issues with balance and equilibrium actually have an inner ear disorder because they are having an issue with one of these two symptoms. Oh, sorry. The other part is static equilibrium. So this is the position of the head when the body is stationary or moving in a straight line. So here in the vestibule, we have the utricle and the saccule, and they detect the position of the head. Inside the vestibule, those otoliths, remember those crystals, remain level on the hair cells when the head is level, causing minimal stimulation. When the head tilts, or when the entire body moves forward, the membrane and the otolith shift, stimulating those hair cells. So you can see here, as this woman bends her head forward, that is moving, and it's causing those hair cells to move, which actually stimulates them and sends an impulse to the brain, telling the brain about the body's head position. So those are kind of three things. You need to know the three parts of the inner ear, what's in each part. Then you need to know what's the three parts of the ear, inner ear, middle ear, outer ear. You need to know all of those. Then in terms of the inner ear, you need to know what is occurring in balance, both dynamic and static equilibrium. And you also need to know how hearing occurs. So you need to spend a little time reviewing those processes. The last part we're going to talk about is vision. So the rest of the slides here will be kind of dedicated to vision and the parts of the eye, a little bit more complicated than the ear. So let's just talk about kind of the accessory structures that we have here. We have eyebrows, so they can enhance facial expressions, aid in nonverbal communication, and really its better job is to help keep sweat out of the eye and shields the eye from glare. Then we have the eyelids. They protect the eye from foreign bodies and block light when closed to allow for sleeping. Thank goodness. I certainly couldn't imagine sleeping with my eyes open. And then they also allow us to blink, which helps to moisten the eye with tears and wash out debris. The papillary fissure really is just the opening between the lids. Then we have eyelashes, which also help to protect the eyes and keep debris out of the eyes. And then we have the conjunctiva, which is the white portion there that actually covers the eyelid and the anterior surface of the eyeball. So it's kind of like one continuous membrane that goes on the eyelid, then comes over the eyeball. It secretes a thin mucus film to keep the eyeballs moist. So it does have a job there, again, of protection. The next structure I want to talk about is the lacrimal apparatus. So this is more inside, and it's not external. We were just kind of talking about those external features. And what this job really is to do is secrete tears. So we have the lacrimal gland, and we have a series of lacrimal ducts, okay? So the lacrimal gland is actually responsible for secreting tears. So they do a multitude of things. They clean and moisten the eye. They deliver oxygen and nutrients to the conjunctiva, but they also contain a bacterial enzyme called lysosome that prevents infection. So that's really important to help us protect our eyes. The lacrimal punctum is a tiny pore which tears drain into the lacrimal canal and then again into the nasolacrimal duct. So that's why when you cry, your nose starts to run, because the tears are actually draining into the nose. There are some eye muscles. So there's actually 6 muscles that move the eye. You do not need to memorize these muscles. They attach to the bony walls of the orbit and to the surface of the eyeball. We have muscles that move the eye up and down and side to side. So we have 4 kind of rectus muscles that do that. And then we have the superior and inferior oblique muscles that rotate the eye to the side and downward, allow you to roll your eyes, which we all know some of us do from time to time. Okay, so on to the layers of the eyeball. So there's 3 layers of the eye, and we need to know what's in each layer and what the job really of each layer is. So you'll have to spend some time with this material. So we have a fibrous outer layer that consists of the sclera and the cornea. Okay, we have a vascular middle layer consisting of the choroid, the ciliary body, and the iris. Okay, so the choroid layer is the middle layer. And then we have the inner layer, which is the retina, the optic nerve, and the blood vessels. Okay, so the sclera, or the outer layer, really is formed by dense connective tissue. Most is white and opaque. Blood vessels and nerves run throughout the sclera. Okay, is the cornea opaque? No, certainly not. It's a transparent extension of the sclera in the anterior part of the eye. It sits over the iris, and it really emits light. It contains no blood vessels, and it's really the first place that light rays are going to go through to get into the eye. The iris is a ring of colored muscle that adjusts the diameter of the pupil to control the amount of light entering the eye. So it does have a job more than just give us all different colored eyes. The ciliary body is a thickened extension of the choroid that forms a collar around the lens, and it secretes a fluid called aqueous humor. Okay, so that's this here. Okay. Then the choroid layer is a very vascular layer of tissue that supplies oxygen and nutrients to the retina and the sclera. So that's really its job. It also helps to prevent glare. The retina is a thin layer of light-sensitive cells. So the retina really is where vision occurs and where the stimulation of the neurons occurs and sends those stimuli up the optic nerve to the brain, and what ultimately allows us to see. So let's talk about the retina. There's a couple places on the retina that we need to be aware of. First of all, it's important that we understand it really lines the inside of the eyeball. And inside the retina are photoreceptors, rods and cones that are stimulated by light rays to produce electrical or chemical signals and send them to the brain. At the center of the retina is a patch of cells called the macula lutea. Okay, this is this section here. Inside there really is this depression called the fovea centralis, and this is where most cones are concentrated, making this area the area that produces the sharpest vision. Rods are clustered along the outside edges of the retina and are absent from the fovea centralis. Medial to the macula lutea is the optic disc, which we see here. Okay, this is where nerve fibers leave the retina and converge to become the optic nerve. Blood vessels enter and leave the area at the optic disc. This is actually what we call a blind spot because there's no rods or cones there. So there's no visual receptors in that area. We also need to talk about a couple chambers, and then we'll get on to how do we actually see, right, and the processes that go along with vision. So we have an anterior cavity and we have a posterior cavity. Anterior cavity here, anterior chamber really is just from the iris. The anterior cavity is divided into an anterior chamber and a posterior chamber, just like I was showing you. So anterior chamber here, posterior chamber here. And what happens here is a clear watery fluid called the aqueous humor really fills this anterior cavity. And its job really is to nourish the lens and get nutrients to that area of the body. The lens is a transparent disc of tissue just behind the pupil, between the anterior and posterior cavities. The lens is what changes shape to allow us to see for near and far vision. The posterior cavity is filled with vitreous humor. So aqueous humor is very liquidy, very watery. Vitreous humor is a jelly-like substance that keeps the eyeball from collapsing. That's here in this posterior cavity. And so that's important because if we have damage to the eyeball and some of this falls out, the eye is actually going to collapse to some degree, and we're going to get what we call retinal detachment. So the retina actually comes away from the choroid layer and we lose vision. What also happens is the ciliary body here is actually what secretes the aqueous humor, and this fluid flows from the posterior chamber through the pupil here, okay, into the anterior chamber, and it drains into a blood vessel called the channel of Schlemm. And so this is a continuous circle here. That's what they're kind of showing you with these arrows. So now that we kind of know the anatomy of the eye, let's talk about the physiology of vision. We'll spend some time, these last couple slides here, really talking about those different processes. So what happens is light focuses on the retina and produces an upside down image. The rods and cones convert the image to nerve impulses, and impulses are traveled to the brain. So that's, the basic sense. For a retinal image to form, though, these rays must be bent or refracted so they focus right on the retina, and we're going to talk about that. The light rays have to strike the center of the cornea so they can pass straight through, whereas light rays that strike off center where the cornea is curved are bent towards the center. So if they go straight through, then they should be okay. They're only slightly needing to be refracted. However, if they go off of the corner or the edge where the eyeball is more rounded, they're going to need to be refracted farther. And let me show you a picture of what I'm really talking about here. So these light rays come in. The cornea is kind of the first thing that refracts light. So you can see how they slightly have to refract or bend to get there. If they go straight through, straight shot. If they're not going straight through, they have to be refracted or they have to bend to get to focus right on the retina. That's what allows us to really see clearly. Convergence, on the other hand, is a different thing that happens. So proper vision requires light rays from an object to fall on the same area of each retina. So in order for us to see, these light rays have to fall in the exact same place of each retina. So light rays from distant objects are almost parallel, meaning the eyes require little adjustment, as shown by this figure on the left here. Right? We don't have to adjust them very much to get them to fall in the same place because we're looking straight on. However, that's not always the case. When we're looking at close objects, we need the eyes to move more inward or converge on the object to align the visual axis with the light rays. So you see how these eyes have to actually turn in a little bit for us to get the same point into both eyes and to land on each of the same spot on the retina. If the rays or the eyes fail to converge the light rays, they'll fall on different parts of the retina and that will result in double vision. So if people have double vision, sometimes that's an impairment with eye muscles so that the eyes actually can't move. Constriction of the pupil also plays a role. Hopefully we've all seen this on ourselves or on others. And this really helps to reduce blurriness when focusing on a nearby object. The pupil constricts to screen out peripheral light rays. This constriction and dilation really depends on the muscles inside the iris. So the intrinsic eye muscles actually help to move the iris. So the pupil doesn't change. The pupil is just an opening, okay? What changes is actually the iris, okay? So it can narrow the pupil to admit less light, or it can widen the pupil and admit more light. So when we go into a dark room, what do you think happens to our pupils? Well, they become large so we can get as many light rays in as possible. We go into a very light room and our pupils are going to shrink significantly so that we can not be overwhelmed with all of the light that's coming in. Another piece I want to talk about is accommodation of the lens. Okay, so the cornea actually refracts most of the light rays entering the eye, but the lens is there to help fine-tune the rays for sharper focus. So we really can't be without our lens. And we have artificial lenses, right? Contacts, glasses, things like that help us if our lens doesn't work properly. So the curvature of the lens changes to allow the eye to focus on a near object, and that's what the actual definition of accommodation is. And we can see it here in these pictures. So what's happening is we get the muscles to either relax, okay, and expand here, or we get the muscles to contract, okay? So the nearly parallel light rays from distant objects, there in that first picture, require little reflection, okay? The ciliary muscles relax and the lens flattens and thins, okay? However, when we're looking at nearby objects, they require more refraction. So the ciliary muscle surrounding the lens contracts, narrowing the lens, causing it to bulge into more of a convex shape and thicken, which gives us more focusing power. We can refract those light rays more by changing the shape of the lens. When we talk about disorders of the eyes and the ears in med-surg, you'll talk about the fact that the lens sometimes is the issue and sometimes it's the muscles. But often it's the lens, and we then need to wear eyeglasses or contacts to accommodate that. Talk just a little bit about the photoreceptors. So those are the rods and the cones that are located in the retina. And you do need to understand the differences here. So we have the rods. They're at the periphery of the retina. They're active in dim light. They're responsible for night vision. They cannot distinguish colors. Versus the cones, concentrated at the center of the retina, active in bright light, responsible for sharp color vision. So those are the big differences there. We have a picture here from your book that kind of shows this. We get lots of those kind of cones concentrated there in the center, and then we have rods located in the periphery. We want the cones to be in the center because that's where most of those light rays are going to be refracted to, is right in the middle of the retina. And the cones are, again, responsible for our sharp vision or our color vision. So it makes sense that we want those to be in the area where most of the light rays are going to be refracted to. The last piece I want to talk briefly about is the transmission of impulses. So once we get the light rays onto the retina, right, the place where our rods and cones are, which are our sensory receptors or our photoreceptors for vision, how do we actually get them to the brain? You don't need to remember these steps, but what you need to know is that these stimuli leave the eye via the optic nerve. Half the nerve fibers cross to the opposite side of the brain at the optic chiasm, okay? And that's what actually allows us to see the same thing in both eyes. So this is, you see here, this green, if you follow it in, is coming here, and you get some from this eye, some from this eye going to this side, some from this eye on the red going to this side. And so we get, and we're able to see the same exact thing on each side.